Hello class, Mr. Ullman here. Today's topic is meiosis. Now meiosis, if you look at it simply as a series of steps, is not that complicated. Prophase, metaphase, anaphase, telophase, repeat that. The reason why we need to explore this topic a little bit more is based on how meiosis fits into the larger picture of life, and most importantly, how meiosis fits into natural selection and evolution. Meiosis as a process only exists in multicellular eukaryotic organisms. And for that reason, there has to be some greater picture as to why it is that organisms like us will bother undergoing this process. Now, sex is highly varied when we look at one species to the next. We have life cycles that go back and forth between haploid and diploid. Now, a haploid cell, or haploid version of an organism, is one in which it has only one set of DNA. So we refer to it as the N condition. When it's diploid, di meaning two, that means that it has twice the amount of DNA. So a haploid organism, or when it has a haploid amount of DNA, will have only one allele per gene, whereas the diploid version is going to have two alleles per gene. Now, depending on the type of organism, they can go back and forth at different stages. For animals, we're going to exist as diploids almost the entire time of our existence. Only when we form gametes, which could be sperm or egg cells, do we have the haploid condition. For many types of plants and fungus, they actually will rotate back and forth or spend a pretty long period of time in the haploid version, and it's really the diploid version is only going to be present when reproduction is occurring or when it's about to occur. And so some of the species you see here will primarily clone themselves in order to reproduce and create a next generation. Reason for meiosis has to do with amount of DNA, because if you have a haploid cell, which could be the ovum, you have another haploid cell, which could be a sperm. When those are combining together to make a diploid cell, you're going to get the normal 2N amount of DNA. And that N is different between different species. Humans, we have 23 chromosomes, so when we are 2N, each of our cells then has a total of 46 chromosomes overall. So the purpose here in meiosis is to be able to make sure that we can produce those haploid cells. We need to take our 2N amount of DNA, divide that in half, so that when the cell from one organism, from a male, will combine with the DNA from a female cell, then we're going to end up keeping the amount of DNA constant from the parent to the child generation. Because otherwise you're going to end up multiplying exponentially the amount of DNA, and that's just not going to be able to fit inside the cell. So meiosis is reductive for that reason. It's going to be, the purpose is to reduce the total amount of DNA. Now when we look at the chromosome, there's a little bit of terminology and language that's a little bit difficult sometimes at the beginning to be able to get a hold of. And so one of those is the idea of the sister chromatids. So a chromosome, every cell is going to have two copies of that chromosome. One of those chromosomes comes from your mother and one comes from your father. Now, during the process of meiosis, that amount of DNA is going to double. So instead of having one from the mother, one from the father, we're going to have copies, so there'll be two from the father and two from the mother. The pairs that are identical are going to be called sister chromatids. When you have that set all together, then you're going to get those homologous pairs. So if you take a look at the picture here, you can see what's in the box here. That is a homologous pair, or a homolog. That homologous pair is really made up of four parts. We have a pair of sister chromatids that are coming from, let's say, the mother, and another pair of sister chromatids coming from the father. So altogether we get 1, 2, 3, 4 sets, and so altogether that would then be 4N. So we start out, again, meiosis, replicating DNA, so we're going from 2N, one copy of each, to being 4N, two copies of each. You can see that again here. If we have non-sisters are represented by different colors here, so the red and the blue, but then the sisters remain paired together. So we have the pair of sisters in red and another pair of sisters in blue, and then all together we call that a pair of homologous chromosomes. Okay, another example here, and looking at just meiosis in the most briefest of senses. We have the blue representing the paternal, red representing the maternal line, and so each of those chromosomes will double. So we start out with a 2N cell. The chromosomes double, so we have 4N. Then we go through meiosis 1. We're going to get a split there. So we have, again, the back down to 2N, and then we go through meiosis 2. Those will split again, and so we end up with 4 cells in the end. Each of those is going to be haploid, being just an N set of chromosomes. So when we look through some stages, so first of all, you do need to be aware of the names given. We have really 8 stages here in meiosis. Each of those is going to repeat with a lot of overlap in terms of what happens in each of those periods of time. Prophase, metaphase, anaphase, and telophase. And very generally, prophase is when the chromosomes are going to form. Metaphase is when those chromosomes will line up across the center axis of the cell. We have some microtubules and some spindle fibers that are which are part of the cytoskeleton and help to ensure that those chromosomes stay in place. During anaphase, the chromosomes are going to separate and move to either side of the cell. And then during telophase, the the nucleus is going to pinch and divide into 2, and then the cell as a whole simultaneously is going to pinch, and that's going to be the cytokinesis part. In the second set of meiosis, the same steps are going to occur, the prophase, metaphase, anaphase, telophase, with the chromosomes following those same basic steps. Now, a couple points I wanted to highlight here with a little more detail. So one of those happens during prophase 1. During prophase 1, when the chromosomes are all condensing and moving towards the center of the cell, the homologous pairs are going to physically connect. So you can see here that this is a transmission electron microscopic image. So this is this actual image of a chromosome, and there's a little connection here between those homologous pairs. And at this point, the DNA from one chromosome can actually switch with the DNA from the other chromosome, so that we can actually get a swapping of DNA from one chromosome to the other. With a little color, it's a little easier to be able to see that. So if we have, again, the non-sisters would be the red and the blue, so those are coming from different lines. They contain different genes, different sets of alleles for those genes. So at that point where the homologous pairs connect, then we can get that switching happen. So you see the red and the blue switch. So you can see that the end result here is that you have these chromosomes in the middle here have a little bit of red from the maternal and a little bit of blue from the paternal chromosomes that we started with. So in the end, you see that there's not just 2 possible arrangements of genetic information, but you see here actually 4 different arrangements. 100% from the paternal, 100% from the maternal, or a mix of one with the other. This is going to create recombination. So recombination is when we take DNA from 2 different sources, in this case the maternal and paternal chromosomes, and recombine those together. When those sisters separate from each other, it's random which direction they're going to go. So as you can see here, again, we're using the color to represent whether they came from the maternal or paternal line. So you can see the red here, a number of those reds moving up towards the top, and some of the reds are then also moving down towards the bottom of the cell. Likewise, the blues, you have some of the blues moving up and some of the blues moving down. And this is another important term describing what's happening during meiosis called independent assortment. Independent assortment means that the chromosomes are going to move randomly into one of any of the 2 or any of the 4 resulting cells. And because of that, we're going to get a mix of the total amount of paternal and maternal chromosomes that end up in that particular sperm or egg cell. Both those things are important. I'll come back to that in a couple of minutes. Important thing that unlike what happens before prophase 1, when there's DNA replication, between these 2 stages there is no DNA replication. So we're at 2N at the end of meiosis 1, and we remain at 2N moving into meiosis 2. This is where we're going to get the reduction happening and get down to that haploid amount of DNA. So to summarize, we start with those pairs. The pairs are going to duplicate to create these pairs of sisters, so we get these homologous pairs. We're going to then separate those during meiosis 1 and separate them again during meiosis 2. So the purpose here, we create haploid cells, which is important, and then we create cells with unique combinations of genetic information. Two main ways in which those unique combinations are going to be made, which I've already mentioned, is independent assortment and crossing over. So what do we know about sex? So sex increases variation, and it increases it exponentially. And so just a little bit of math here to be able to emphasize that point. And so when we look at that independent assortment that's going to happen, because each homologous pair has a 50-50 chance of lining up so that you get mom or dads of that particular chromosome into a gamete, that means that there are two possibilities for each of the 23 sets of chromosomes. So if we take that mathematically, 2 to the 23rd power, we've got over 8 million possible combinations of chromosome arrangements per gamete. So again, this is independent assortment, the fact that for any given sperm or egg cell, whether it gets the maternal or paternal chromosome, that you have an equal chance of either. Now, fertilization happens randomly, and so we've got those 8 million plus possible combinations, and each of those has an equal chance of combining with any of the other 8 million possible combinations that are occurring in the gamete of the opposite gender. And so we multiply those by each other, and we're going to get, 70 trillion possible combinations per offspring. When we look at that, I mean, this gives us a great illustration of why it is that while siblings share many similar characteristics, why siblings can look alike, while that there are still going to be an obvious number of variations that are going to happen even between two people who are very closely related. Two siblings who have the same mother and the same father, but because the DNA is going to end up mixing in a different way each time in terms of which alleles are passed on by the father and which alleles are passed on by the mother, that explains why we get that mix in the offspring. Now, we add into the fact crossing over happens, and because crossing over happens and we get that little bit of extra mixing that happens so that within one of those chromosomes that's passed on, it can have both mom and dad DNA in it. There's not a mathematical way to represent that, but ultimately it means there's an infinite total number of possibilities when we factor in the fact that it really could be a combination of both of mom and dad in a particular chromosome in a particular gamete. So this ends up being really important in terms of then being able to apply this to the idea of variation and then selection. So being able to mix up your DNA allows for more variation in your offspring. So if you have more variation in your DNA and you have unique combinations of alleles, that means that you're not going to remain stagnant in terms of the total gene pool. You're going to have the potential to have a novel mix of DNA in the next generation that might end up being beneficial towards the survival of that offspring. So when we look at a population, the more variation you have, the more fit the population is going to be, particularly when the environment changes. It is in cases when there is low variation, then we get a lot of inbreeding, we get a lot of the expression of a lot of recessive traits, of unfavorable diseases that then really hinder the health and the reproductive success of that species. So things like the Florida panther, where the number of them is so small and they're so genetically identical, they're nearly genetically identical towards each other, and so they're going to end up being less healthy than a population that has a wide range of possible DNA. Last few topics here. One is some analysis of chromosomes that we do. So when we look at a karyotype is going to be a picture that is taken using an X-ray of an individual's chromosome. So it's really an X-ray through a microscope. So here's the technician being able to take that picture. That picture of kind of the scrambled chromosomes needs to be then cut apart and matched up so that we get pairs of chromosomes all sorted together. When we look at, they're sorted in order based on size. So chromosome 1 in humans is the largest in size, chromosome 22 is the smallest in size, and then the final set of chromosomes, chromosome number 23, those are going to be the sex chromosomes. So the X chromosome is actually larger than many of the others. However, we always put the sex chromosomes at the end, just based on tradition. So one thing that you need to notice is the difference in terms of the size. The X chromosome is large, the Y chromosome is quite small. And this then becomes important in understanding how a lot of sex-linked traits are inherited. There are a lot of genes on the X chromosome, and everybody has an X chromosome, both male and female, but the Y chromosome has only a small number of genes that really are only there to impart particular male characteristics or male features. So X is shared by everybody, but Y is going to be just for males. And so then you can see in a female human karyotype, you see the pair of X's here. You see the X and the Y, and they look different from each other for the male. Non-disjunction is one of the defects that can occur during meiosis. It can happen during anaphase, during meiosis 1 or meiosis 2. Anaphase is when the chromosomes need to split and move to either side of the cell. And so if those chromosomes stay stuck together, and so they don't split evenly, you have more chromosomes moving to one resulting gamete than another. So if you have monosomy, in this case you've got only one copy of a chromosome. In most cases this is going to be lethal. So if you have, say, chromosome 1, if you have only one copy of an embryo is never going to develop. That fertilized egg is not going to ever turn into an embryo that would turn into a fetus. One counterpoint to that, though, is Turner syndrome, in which having only one X chromosome, that's an example of one case in which it is not lethal. Trisomy is more, not necessarily more common, they're actually equally common to each other, but trisomies are a little bit more well known because, particularly because of Down syndrome, which is fairly common. Here you can notice Down syndrome is trisomy 21 because there are, instead of 2 copies, there are 3 copies, so there was an extra copy of chromosome 21 that was a result of a non-disjunction event, and this is not as lethal as some of the other monosomies and trisomies. In this case, having that extra DNA still, though, is problematic, and so we see some of the, as you look up any of these particular non-disjunction events online, you can see what some of the complications and see what some of the symptoms are of having that extra set of DNA. You'll notice that Down syndrome is more survivable because chromosome 21 is quite small. Small chromosomes contain less genes, and so there's less overall effects as a result of having that extra DNA. Okay, and that covers what we're going to look at today in meiosis. Thanks for listening.